So what I thought I'd talk about this year was really just to have some slides that would be a good kind of catalyst for discussion rather than give you a lecture. Because I think for the last 5 or so years I've given you sort of what we think these various diseases are, and I think most of you have heard that already, and certainly we can go back and talk about some of those things. But mostly, as Ken and I prepare for this trip every year, we say, okay, where are we on Ron's clock? So we've made it to Cloud's Rest, right? We're taking our pictures and ready to head back down. So we're 5 years into the 10 years we've been given to try to help solve these diseases with your partnership. And I think what I kind of wanted to do was just sort of be transparent about what our process is, about where that's gotten us, and what support from HistioCure has done so far. So mostly it's just going to be kind of one-shot slides of general topics that can hopefully be a springboard for conversation. And I can be cut off any time, because I know last time I went over quite a bit. So, all right, so I don't have to tell you why these diseases are an issue. For LCH, what are the major challenges? So, a lot of times I think LCH, because of its ambiguity as a cancer or inflammatory disease, has maybe not gotten the same kind of gravitas when discussing it with physicians or the scientific community. But in fact, we know that LCH is just as common as Hodgkin's lymphoma in children, and actually the outcomes are just about the same, if not worse, in LCH. And not only is survival kind of similar, but we do a much worse job. So what we consider standard of care only cures 50% of children, and the kind of designed to improve that standard of care are these trials that take up to 5 years to really get an answer on. And so I think that pace of progress is suboptimal, and clearly our outcomes are not acceptable. And kind of going along with this is not only do a lot of children not respond, there's a very, very high rate of relapse. And we know that children who relapse more and who have persistent disease suffer complications from both the therapy they get as well as having persistent disease. And I think there's been some debate in our community. Even now, there's differences in practice about if someone has just a touch of LCH, do you let it ride? Is that okay? Do you treat it symptomatically like an autoimmune disease, or is the goal really to cure it? And I think probably one of the biggest messages from the research we've done so far with our HistioCure support is that our bias is that you have to cure LCH. I mean, you don't want to cure it with a sledgehammer if it only needs a tickle, but I think it's important to try to cure the disease. And then neurodegeneration, I think, is a thing that hangs over the head of all patients and families who have children who are cured. You say, is this coming down the road, and how can we understand this process better? If there are signs of it, how can we address that? And Ken's touched on some of that. And then, of course, HLH is a little bit less nuanced on why one needs to come up with better therapies. I think that there's been huge progress in the last 30 years in people discussing HLH, recognizing HLH. But now the pendulum's kind of swinging to where everyone's seeing HLH everywhere. And in some ways, it's I think, great that people are thinking about it, but I think our work now is to really refine it. Because HLH is a syndrome of extreme inflammation. Some kids don't need etoposide and dexamethasone and bone marrow transplant, but some kids do, and they need to get it quickly. So how do you differentiate who needs what? And then adults, it's incredible. The adult community has started checking ferritins and then led them to look into sort of immune activation, and they realized that this is actually a huge problem in the adult community. In fact, we have colleagues at MD Anderson who we're working with who say that now they're looking for it. They see it in a huge number of their patients with myeloid malignancies. And I mean, it's not the same disease that kids have, but this problem of the immune system being revved up too high, resulting in organ damage to that patient, is, I think, a common theme. And so how do you dismantle that reaction is a common challenge. And then, of course, long-term complications from therapy. The therapy that used to treat HLH is pretty extreme and sometimes can result in problems. And then this sort of the weird stuff. Here's LCH and HLH are the diseases that get talked about a lot, but what we're realizing now is not only are there different conditions such as Rosai-Dorfman or Time-Chester disease, these other things that have their own names, just about everybody has their own version that's sort of a hybrid of some kind of LCH or HLH, and everyone, this is really sort of a group of diseases where everyone has a personalized version of the disease, and so we need to come up with personalized approaches to manage it. In general, how do we do better for this entire group? So I want to make this interactive. So Ken, do you agree? Do we agree on that? Okay. So any questions on that, the sort of big picture, general concept from the group?
You had some comments of success rate, that only 50% someday cured. But my understanding was the fatality rate had decreased dramatically.
Correct. So, but I think it's not a mission accomplished.
I agree.
Right.
We're still here.
Yeah. So if you go back to that last slide, Ken.
But so the fatality rate, so what I tell people when I'm fundraising, let me know if I'm saying this accurately, is it used to be like 55% fatality rate, and now it's almost not measurable, it's almost zero?
For LCH?
For LCH.
So I think in the LCH3 study still had about 10-15% mortality. So it's not.
Okay.
It's not. The problem's not completely solved, but it's true that because people are recognizing the disease, and I think some of the approach is a little more standardized and supportive care is better. The outcomes are slightly improved, but if you look at the therapy between LCH2 and LCH3, it didn't really change at all with that difference in outcomes. So I think some of it is just a difference in supportive care.
And also the second line therapies. So
Right.
a decade ago, people were only using 2-CDA, and that worked some of the time, but not very much of the time. And before that, people just keep using the same drugs over and over and over. Now we have some new combinations of things like clofazimine that works really good, and so the mortality rate's less, but it's still.
Right, that's a good point, Ken, because when people are measuring, for example, on the LCH3 trials, when they're measuring survival, it's way beyond the time you're actually getting the initial therapy. So the question is, what happens to the kids who fall off the study and end up getting other treatments downstream? And so that's not really been tackled in an organized fashion.
10 or 15% mortality rate, does that apply to people under the Ken and DrCarl standard of care, or under your?
I mean, that's the prospective study. I'd say we haven't. So we see about 100 new cases of LCH a year, and I don't think there's been a patient at TCH who's died in the last 5 years or so.
Yeah, so that's the spec I use, which people find really impressive. This fatality used to be 55% in patients under our care. We haven't had anyone die in years.
Yeah, and even the patients who come to us from elsewhere and then go back to get their therapy elsewhere, I can think of only one fatality.
All right, so you guys are the history of wizards. So
Well, our goal is to make everything we do sort of generalizable. So you don't have to come to Texas for care. And obviously, there's other people doing good work as well. But I think there is a lack of, other than people who are sort of specifically interested in histiocytosis, I think there's not a lot of good resources for people to figure out what to do. So. All right, so getting into this a little bit, the history of histiocytosis research, so this really been sort of the rare disease model. And I think the rare disease model is an effective one in a lot of cases, but I think lumping LCH into 1,000 other diseases that can go through the Office of Rare Diseases at the NIH, and everyone sort of tries to get some bit of attention is maybe less productive than if you can figure out what is LCH specifically and put it in its family, and then even though it's rare, well, medulloblastoma is rare, Hodgkin's disease is rare, but if you look at it as pediatric cancer and we call LCH pediatric cancer, then I think there's access to more resources. HLH certainly arises in the setting of cancer, but it's really more of an immune disorder. But it's interesting now the immunology community is really embracing HLH quite a lot. And there's a lot of really prominent people across the globe who are leaders in thinking about immune disorders, which historically have been absence of immune function, severe combined immune deficiencies, things like that. And that's really been what people focus on. But now I think they're recognizing HLH as part of this spectrum where it's immune dysregulation. It's just that it leads to immune activation. So there's a huge amount of attention recently on HLH globally, and I'll kind of explain some of that as I go through the talk. Okay, so both HLH and LCH, I think, can attribute a lot basically where they are now and a lot of their success, and that anyone is paying attention to these diseases, to the efforts of the Histiocyte Society. People started getting together and comparing notes, and that's really the first step. But one limitation is it's a group that's not funded particularly well. It's got sort of anything international is harder to do than an institutional trial or multi-center trial. It's about 5 to 10 years per study. In addition to Histiocyte Society, there have been some national research efforts. So Japan has a national histio group, France has a national histio group. I'll talk a bit about, we've really created one in the US with the North American Consortium for Histiocytosis, and the Children's Oncology Group is taking on histiocytosis. Those are some of the advances I'll talk about in a bit. So I think in 2010, when LCH gets branded as a cancer, this really opens up some new opportunities, even though for some reason, this month, there's some debate in pediatric blood and cancer, about this maybe being an overstatement. I don't think it really matters. If we can take advantage of this, then I think that's particularly helpful, even though it may carry some sort of connotations or create some prejudice in the way people think about the disease if it's their child. But I think that LCH is LCH, whether we put it in this family or not, doesn't impact how the child's going to turn out. Okay, so let's talk about our experiment with the Texas Children's Histiocytosis Program, which really began 25 years ago with Ken sort of being interested, getting patients in the door, then creating sort of a critical mass of clinicians interested in the disease, this disease group, to now we have, and I'll show the slide of our growth. I think we have about 20 people or so in our lab. We have 4 clinicians who are dedicated to LCH and HLH. And it's the busiest clinical program at the largest cancer center in the country. So I think this model can work, and I think what the point isn't for everyone to come to Texas, but I'm going to kind of demonstrate how some of the things that we've been able to do with your support has been leveraged into sort of national and international progress. All right, and so this is sort of the, I like to channel Bruce Lee sometimes when I'm trying to be effective. And I saw this this quote, and I thought, yes, this is a little more interesting than Malcolm Gladwell's. You have to do something however many hours, but it's I fear not the man who has practiced 10,000 kicks once, but the man who's practiced one kick 10,000 times. And I think there's something to be said for that. Certainly, you want some people who can do lots of kicks, but in some situations, just being good at one thing can be equally effective, or even more effective. Okay, so how do we make improvements on these diseases? And these are some of the things that we talked about, when we got together with all of you over the last decade or so. How can we improve outcomes for our community? So one challenge is that there's a small patient volume. So an answer to that was to really kind of create a dedicated program. Another challenge is that it's really difficult to get tissue specimens for research. They're almost non-existent. But to answer that now, everyone comes, having a large volume, we try to also offer everybody an opportunity to participate in research. So you come, it's not required, of course, but I think most people are motivated, and over 95% of our patients are enrolled in research. We have over 1,000 people on our histio studies now. Additionally, we have community research opportunities. So with HistioCure, and I'll talk about Spit for a Cure in a second, we've really been able to get this national and even global outreach where people are sending us spit samples, and we can do a lot with those spit samples, but even more what we can do now, for example, we're doing a study where we're trying to figure out what are the gene mutations in every patient with HLH, what are the gene mutations in biopsy samples from every patient with HLH. We can not only sequence the spit, but we can call their pathologist and say, hey, send us the blocks. We can call their doctors and say, hey, can you send us a blood sample? And so it really lets us take advantage of new things that arise without having to go to the cost of getting everything at once in the freezer. We have access to the patient, so we can go back and ask for participation down the road. And so we have a research team dedicated to doing this. It's not a minor effort. I mean, it all kind of started as a me and a centrifuge, but thank goodness now we have about 10 people involved in this process. All they do is get tissues kind of collected appropriately with high quality. So another problem is that once you get tissues, what can you do with it? And luckily technology has come along where you can get a huge amount of information even from just a paraffin sample that we couldn't do 10 years ago. But not only that, we save samples in a way that we can kind of take a single cell out and ask a question, what does the gene expression of a cell that has one special phenotype compared to another, how do they differ? And then there was no money, so you're helping us solve that. And then the funding from HistoCure has really has helped us to generate the preliminary data that's led to grants. And so since we're talking about Spit for Cure, I thought maybe right now we could show the cartoon. You can see I've got some creative people with maybe too much time on their hands every once in a while, but we'll show you this cartoon that hopefully we'll put up to motivate more people to join. This is how it all works. So my team got tired of explaining this, so hopefully we can just put it on the web and then.
We're prior to this cartoon, were people getting confused?
It's still coming, yeah.
Oh.
Now it gets kind of serious. We got to change this music. Yeah. So that Brooks was our lab manager. Maria is our research coordinator, and that's Ernesto, who handles the samples. And that's Alice, who's a student working with us, and Tommy's a MD PhD student working with us. So that's Aaron Peckham, who's the epidemiology postdoc who is working on the project with another junior faculty, Rikia Chakraborty. So let me just get rid of this. So, but in addition to the LCH study, which I'll talk about the results of that in a second, we also are doing that with HLH, and so we're working with Jordan Orange, who's the head of immunology at Texas Children's, and we've sequenced 100 kids with HLH now, and interestingly in Texas, only about maybe 20% have previously identified HLH mutations. But another, I say 30 or 40% have mutations in genes that impact the immune system that haven't previously been associated with HLH. So I think, even though if you read the textbook, everyone has mutations in a specific pathway, I think you can kind of arrive at this place of extreme inflammation from a lot of different routes. And so that's, I think, another great use of the study. Okay, go ahead, Ken, for the next slide. So again, this needs to be updated a bit. I think we've sort of plateaued a little bit. We're getting about 150 to 170 patients a year kind of locally enrolling in our study. And I think that's thanks to this group. And so the way this works is this line is the patient census at Texas Children's who have ALL, which is the most common pediatric cancer. And you can see this slowly is rising with the Houston population, but not anything like this curve. So this probably doesn't represent a new epidemic of LCH, but this is LCH, HLH, and everything else. But increasing referrals, but also increasing awareness from our referral base. So again, the model is we get everyone in the front door, we get everyone enrolled on study, we try to collect tissue samples, try to study those tissue samples, and then see how we can kind of make improvements. And I think for the first 3 or 4 years or so, we were focusing kind of in this direction, but we're really now getting to the point where we have quite a lot of clinical trials baking, where we can take some of those discoveries and complete that loop. And then the idea will be we keep kind of making refinements and using this model to improve outcomes for our patients. And this is my Ron slide. I say, well, what does an engineer think about? So this is LCH or HLH, and the problem is one part's broken. So how do you figure out? And is it the same part for every patient, or is it a different part and sort of a common just in the engine, or how does it all work? So really, our first focus was to kind of deconstruct the biology of the tumors or the immune system in these patients. And then we subject them to all these different kind of studies. So looking for germline mutations, the genome-wide association, which we just talked about, looking at least in LCH and other related diseases. Are there mutations in the tumor that have been acquired that are different than inherited mutations? How are the cells functioning? How are they behaving with each other? And can we use this clinically? Can we take some of these findings and look and see, how are kids different or are kids the same based on some of their biomarkers? For quality, how are we doing with our outcomes? Are we having consistent practices? And then moving forward, what's the best way to make sure that people stay healthy? And so in addition to having over 1,000 blood samples, we have over 500 frozen LCH samples in 10 years, which is really an unprecedented resource. And again, when we get your blood samples, what we do is we kind of spin it down, and this yellow part's the plasma, and then this is the cloudy bit here is actually where all the cells are. And so from the plasma, we're able to look at all the plasma proteins. We're able to kind of find DNA within the plasma that tells us, is there the BRAF? That's where some of the BRAF assay comes from. And from the white blood cells, we can find, what are the immune cells doing? In the case of LCH, is the LCH precursor there? We can collect genomic DNA. So we can do a lot just from a simple blood test. And so the idea is we kind of look at, we can kind of take these things and then test them in preclinical models. There's two kinds of preclinical models. One is when we introduce the same disease, can we create an animal with the same disease? So for example, we've been able to create a mouse that gets LCH. And this is in collaboration with Mira Murad's group at Mount Sinai. And so when we overexpress this BRAF gene at a specific stage in differentiation, these mice basically get high-risk LCH, and this has been really useful for us to see what are the differences in these mice, and then we can go back to our human samples and say, do we see the same differences in our human samples? And hopefully we've got a paper coming out soon that really kind of explains some of the mechanisms of why LCH arises. And then this is an example of what we can do just from a frozen tumor sample. So if we get a frozen tumor sample, we can run it through, for example, this is called an image stream cytometer. So each one of these things is a single cell. Okay, and so each of these cells, they say, well, how much MEK are you expressing? How much CD207 are you expressing? You can see none of them are CD3, that's a T cell. And you can kind of put all of these equations together to say, okay, well, when we have these tumors, so this patient who has a BRAF V600E mutation, they respond to a BRAF inhibitor and to a MEK inhibitor when we test this just in the Petri dish. Patients with this particular mutation, the MAP2 kinase mutation, won't respond to a BRAF inhibitor, but do respond to a MEK inhibitor. And then these patients who haven't been characterized, have sort of random responses. And really where we can go with this is eventually to say, well, we can just get a patient's tumor and just test a bunch of different drugs and predict how their tumor is going to respond, even if we don't completely know what the mutation is. So these are some preclinical models that exist that didn't exist before. Go ahead, Ken. And then also, like I mentioned, this isn't just us doing everything here, but with our tissue resource, we're able to leverage expertise from people around the world. So we have active collaborations here in the Bay with the San Francisco and UCSF group, with Miriam at Mount Sinai, Matt Collin in Newcastle. We also have a collaboration in Singapore with Florent Gino, who's one of the leading immunologists in the world. And we now actually are expanding, and Texas Children's is expanding into several sites in Africa, and we're looking at, well, what does histiocytosis look like in Uganda? So Joseph Lubega, one of our faculty, has started a fellowship program there, and we're actually going to have about between 10 and 15 sites in Africa in the next probably 10 years or so. Okay, so all of this science is nice, but knowing what's wrong, not really why you're here. You want to have, be able to fix the problem for your children or for at least the next generation. So we have some clinical trials, either in development or actually active. So Olive may talk. Is Olive here yet? No. So one of the things we discovered was that we think that LCH is caused by the sort of myeloid precursor. It's sort of closer to the acute myeloid leukemia than maybe it is to other kinds of lymphoma. And so if we take some of those myeloid drugs, could they be more effective than vinblastine and prednisone? So we have a randomized trial ongoing right now. We have our first 2 patients enrolled, testing vinblastine prednisone versus cytarabine. And with 100 patients, we should be able to redefine the standard of care if our guess is right. And as soon as we get approval from our regulatory group, this will open in UCSF and Stanford and about a dozen other sites around the country. And this is funded partially by the HistioCure group. So we're talking with a company called Plexicon about testing a second-generation BRAF inhibitor. That's not open yet, but we should be able to I think that's going to move forward, and we're at least doing preclinical testing with this new agent that hasn't been tested in children in any disease yet. The North American Consortium for Histiocytosis is something that's grown out of some of our collaborations. So Carlos Rodriguez Galindo, who's at St. Jude's now, and I put in for a grant to St. Baldrick's Foundation, which is a childhood cancer grant. And they have these specific grants to fund consortia. And so it's a $2.5 million grant over 5 years. And there's 11 centers that have agreed to collaborate on development of clinical trials. So basically, we're taking our Spit for a Cure model and applying it to these 12 different sites. And the interesting thing is now, since this got started, not only is there the initial 11 or 12 sites, but we've gotten inquiries from another 35 sites that want to participate. And so I think it really speaks to the need to have sort of a consortium. And the point of this group is to do the early phase studies, phase 1, phase 2 studies that can then lead to the bigger definitive trials. So there's also a study testing the efficacy of clofarabine. As Ken's mentioned, we think clofarabine works better for salvage. That study's open now at several sites. And then Nacho Libre, this acronym makes sense. I forget how it makes sense, but it actually does. And this is just a retrospective study to look. Everyone who's sort of in a desperate case pulled a BRAF inhibitor or MEK inhibitor off the shelf and tried it in a patient. Can we figure out what the outcomes are, just so we can capture the experiences from those patients? So actually, if any of your children have received salvage therapy with these targeted agents, or if you know of people who have, let me know, because we can enroll from anywhere in the country. And then we're talking with Steve Simcoe, who's now at Genentech. They have a drug called cobimetinib, which is a MEK inhibitor. And this is still a concept, but we're working through the possibility of doing a phase 2 trial with cobimetinib with them. So the Blood and Marrow Transplant Clinical Trials Network is an organization dedicated to looking at bone marrow transplant studies. So they decided several years ago that HLH was the biggest challenge they faced, with 70% survival for a non-malignant condition was really the worst disease they encountered. And so we worked to just kind of create a standard of care protocol to see how this reduced intensity conditioning concept developed in Cincinnati would work on the national scale. So we've completed a accrual, and we should have the results coming. And the point of this now is we have a baseline, and we'll be able to make the next intervention and see if we can improve on that. HIT-HLH is a collaboration with Mike Jordan at Cincinnati that we participated in for frontline therapy, and that's completed accrual. And then we're also one of a few sites in the U.S. working with Novimmune, and I'm on the scientific steering committee for that company, to really test this revolutionary concept to block interferon gamma signaling rather than using etoposide for kids with HLH. And so I think that's in the process of undergoing FDA approval, and hopefully we'll have results from the first cohort published soon.